Hello, everyone. Thank you so much for joining us for the third of our official Snake Week webinar series. Really excited to have you all. Snake Week was the brainchild of me, of Project Rattlecam, and Melissa Amarello of Advocates for Snake Preservation, and Christina Zdenek from the Australian Reptile Academy and also the IUCN Snake Specialist Group. We thought that for the Year of the Snake, it would be a good idea for us to have not just one day, World Snake Day, that we have every year, which happens to be today. So happy World Snake Day, everyone. But also an entire week. And so we've organized this and we have dozens of organizations. In fact, I think it's up to 70 organizations now around the world that are holding in-person events and online events celebrating the wonderful world of snakes. And as part of this, we organized a webinar series, and we're so thrilled today to be providing you with this wonderful webinar from one of our favorite biologists. So here's how this is going to work today. We'd like you to please keep your mics muted throughout the presentation. Any questions that you have for Dr. Shine, you can enter those questions in the chat. And at the end, before the end of the hour, we'll have hopefully about 10 minutes or so for questions, and I'll moderate those, and hopefully you can get your questions answered. I will be posting this recording to YouTube afterwards if you'd like to share it with anyone at that point. In the chat, you can please limit your chat to those questions or conversations about Dr. Shine's presentation. So this is the third of our 6 seminars that we've organized for Snake Week. We started with Ram Whitaker from India talking on Monday about king cobras, and then we recently had Rulon Clark talking about rattlesnake hunting. Today, we are honored to have Rick Shine talking about Australian snakes and his efforts in conservation biology. And then we have 2 more tomorrow. We have Jazz Ong talking about king cobras in the Philippines, and Jesus Rivas talking about anacondas, of course. And then finally closing it out on Friday with Hiral Naik talking about South African snakes. In addition to all of these, there's a series of shorter webinars that are organized by the IUCN Snake Specialist Group. And all of these can be found, including registration links, at snakeweek.org/webinars. So without any further ado, I want to introduce tonight's speaker, or if you're in Australia, this morning's speaker, Dr. Rick Shine of Macquarie University in Sydney, Australia. So when I got into snake biology about 30 years ago, Dr. Shine was already kind of the biggest name in the field. He has done research all around the world. There's some websites online that show the collaborations that scientists do, and he's got just dots everywhere, and he's worked everywhere around the globe. But most notably, of course, in Australia. Dr. Shine has, I couldn't even count the number of papers he has. It's somewhere around 1,100 according to the internet, which may or may not be correct. That's an absolute stunning number. And he has 4 books, I believe, according to the internet, which is a stunning number as well. Highly recommend his books, written for a popular audience and telling all of these stories about the many adventures he's had with snakes and the science of snakes. So thank you so much, Rick, for joining us tonight, and you can go ahead and share your slides now and take it away.
Okay. See if we can get this up and running. Okay, I'm hopeful that you can now see the presentation.
Looks perfect.
Happy International Snake Day, everybody, and thanks for including an Aussie among the people that get to talk about their favorite animals. My talk is really about that kind of very fundamental question that any researcher has to ask themselves, and that's what's the point of doing all this research stuff? It's all very interesting and entertaining, and we have great fun doing it, but can we actually bring it to bear on conservation? And sometimes that can be rather difficult to do. So I'm going to give you an Australian perspective, and a lot of it will be just my opinions as to how we can best use research to inform conservation. Okay, that's working. So what's the conservation status of Australian snakes? How well are snakes doing in Australia? What could we potentially do to improve that conservation status? And I'll start by giving you a quick introduction to the snakes of Australia, because it really is an incredible success story in evolutionary terms. We don't really know exactly how many species of snakes there are in Australia, but there's at least 170 or so. When people think of Australian snakes, they mostly think of big deadly things like this spotted black snake down here at the bottom of the screen. But in actual fact, most of our snakes are small. They're harmless to humans, like this little black-headed snake. In fact, 50 of that 170 odd snakes species are blind snakes, typhlopids, that look more like worms than snakes. There's been an incredible increase in the recognised number of blind snakes in Australia when people started using genetic evidence. The damn things are hard to tell apart until you've got genes, but once you've got the genes at work, you discover that there's a lot more types of little burrowing snakes than you thought. At the other end of the size extreme, we've got 15 species of pythons. Several of them get very large, and of course, they're very popular in the pet trade and so forth, much better known to captive keepers than they are in the wild. We've got 11 species within the so-called harmless snakes, the colubrids and the mangrove snakes, the homolopsids, most of them living in trees or in the water. We've got two wonderful file snakes, acrocordids, these incredible slow-moving guys with baggy skins that spend their life underwater and live in the slow lane. In some cases, females, have a litter of babies once every 10 years. More than 100 species of venomous land snakes, the elapids, the group that also includes bombas and cobras and coral snakes in other parts of the world. And so this gives Australia its distinction of the only continent where most of the snakes are venomous. And within the elapids, there are 32 species of sea snakes. The sea kraits, like this guy, they're probably the original ones that came to Australia from Asia, and they gave rise to the Australian elapid radiation on land. And then that radiation in turn gave rise to the true sea snakes that have since diversified at an incredible rate in the coral reefs of the Indo-Pacific. But although I said 170, but we don't really know how many species we have. And I've got two graphs here to show just exactly how difficult it is to put a number on it. This graph with the blue histograms is from the reptile database, and it's the number of snake species that are recognized worldwide. So when I was a student in 1970 or so, I could confidently tell people there were about 3,000 species of snakes in the world. Now I can tell them that there's at least 4,000 species. But if you look at this graph, there's no hint there that it's slowing down. If anything, it's going up faster and faster. So there's probably a hell of a lot more than 4,000 species of snakes worldwide. It's just that we haven't got round to describing them yet. The other graph with the red dots is from Hal Cogger's field guide, the first comprehensive field guide to reptiles and amphibians. The first edition came out when I was a student in the early 70s, 75. There were 650 species of reptiles and amphibians in Australia. The latest edition of the field guide, which is already out of date, has about twice that number. So the number of reptiles and amphibians living in my country has doubled over the course of my professional career. It's extraordinary how we underestimated the diversity until recently, and we probably still massively underestimate it. So I guess if you're talking about snake conservation, for most audiences, the first question is why bother to conserve snakes? The world is full of people like this gentleman on the top right of my screen with a dead tiger snake many years ago, who believe that snakes are horrible and awful and terrible, and you should just kill everyone you see. But the fact that you guys are here listening to a talk about snakes suggests that you probably don't ascribe to that particular approach. So I'm not going to waste much time trying to argue with you that snakes are worth having around, but I'll just point out the sorts of arguments you can make, certainly from an Australian perspective. First is that snakes play really important ecological roles in Australian ecosystems as prey for other predatory species and as predators on many taxa themselves. Medically, we're seeing already many medical breakthroughs coming from the use of snake venoms. I saw a new paper today that was pushing the envelope still further. Economic, snakes play a huge role in helping the farmers because they eat a vast number of rodents. And this little picture down here on the right is from an article in The Conversation last year, where we sort of did some calculations and worked out that really the presence of brown snakes on a farm massively increases the profit margins for farmers. And they really should think twice about thumping snakes on the head, because the snake really is their best friend. Aesthetics, that's a harder argument to sell with a lot of people, but I see phenomenal beauty in snakes. I mean, I think, the Mona Lisa is a great painting. The world would be a worse place if somebody destroyed the Mona Lisa. But really, this was just the work of one guy over probably a few months at most. A snake species has been sculpted by evolution over millions of years. It's infinitely more complex and, to my eyes, more beautiful than anything created by the hand of man. And lastly, there's a cultural perspective. Certainly within Indigenous culture within Australia, with Aboriginal people, snakes are really important cultural icons. And so there is a huge value to conserving them, simply because it helps to maintain the continuity of Aboriginal cultural songlines. So, why are snakes disappearing? We hear a lot about illegal or legal trade. I've worked a bit on this. This is me many years ago looking at a reticulated python in a slaughterhouse in Sumatra. I actually think this is not a big deal for most species. Although the numbers of snakes dealt with in trade are high, in most cases they're species that can handle that pretty easily, and we have pretty good data on sustainability of that harvest. I think habitat destruction is a much bigger problem, and that's happening all around the world. Climate change is turning out to be catastrophic for at least some kinds of snakes, because they are so intimately dependent on ambient temperatures. And feral pests like cats and foxes and cane toads and disease are increasingly emerging as a real threat to snake populations. So they're the kinds of issues we've got to deal with. And I guess the main argument of this talk is that we're not going to understand what's going on unless we have people out there doing that research. That's sort of a statement that every researcher is going to agree with, but a lot of the general public, I think, thinks that a lot of the research is very arcane and specialized, and it's mostly done by researchers to give them an opportunity to, like this picture on the bottom, to go out with a graduate student and catch giant scrub pythons in Queensland. It's good fun. But the reality is that many of the causes for the declines and so forth are actually pretty hard to work out. They're not the obvious ones. And so I think we really do need to understand what's going on. And so research has a huge role to play in driving conservation initiatives. The causes for decline and recovery of snake populations really are complex. And so for the rest of this talk, I'm going to be focusing on three systems that I've worked on, in each case for very long periods of time. The impact of climate change on tropical snakes, the impact of invasive cane toads on some of the elapid snakes, and the enigmatic disappearance of sea snakes from the coral reef ecosystems in Northern Australia. Well, let's start with tropical snakes. Very early in my career, and we're going back to the 1980s here, I found a place up in tropical Australia. You can see on this map, Darwin, the capital city of the Northern Territory. It's a long way from Sydney, where I live, but the tropics are fantastic for a snake lover. The diversity and abundance of snakes is phenomenal. And so I set up a research program up at a place called Fog Dam, not far from Darwin. It's a wonderful place. There's a dam. There's a dam wall that you can drive along. There's a floodplain on the other side that fills up during the wet season when the monsoonal rains inundate the landscape. And it's been a wonderful time watching the landscape, the animals. This photo of a group of people with a snake was a dinner we had with the local buffalo farmers, and a giant olive python turned up halfway through, so we took that photograph with it. So more years of research at Fog Dam than I care to admit. This is me on the left-hand side here, back when my beard was black. Whoop. How do I go back? And This is me with a king brown snake, a larger leopard, back when I was young and brave. This is me now walking the same damn wall, but picking up pythons these days instead of leopards, partly because I'm probably less brave than I used to, and partly because the cane toads have largely eradicated the larger leopards from our system. So what you need to know about the climate in tropical Australia is that it's always hot. The red triangles, the green lights show the temperature, and it's always 30 odd degrees, 100 degrees Fahrenheit, I guess, or whatever. I'm not quite sure how to make that conversion. So it's hot all year, but the rainfall is highly concentrated when the monsoons come through at the end of the year. And they differ enormously from one year to the next. You can have one year with really heavy rainfall early, or you can have a year with very little rain early but lot late, or a year when there's very little at all. And so that turns up driving the ecology of water pythons that were our main study animal. So for many years, Thomas Madsen and I have been looking at water pythons. Here's one eating a dusky rat. And it turns out that the amount of rainfall you get in the wet season is absolutely the driver of everything that happens in that python population. And what happens is that the amount of rain determines the conditions for the rats, the native rats. If the rain is just right, you get a phenomenal amount of breeding of native rats, and so you get a terrific amount of food for pythons. And so Thomas and I put together all sorts of lovely graphs where if you plot the rainfall, you can predict the number of rats, and if you know the number of rats, you can predict the body condition of the pythons, the survival rate of the pythons, the proportion of the females that are breeding each year, and things like that. So this was a lovely story about how climate drives prey populations, which in turn drives the biology of the snakes that depend on those prey. And we were feeling, boy, we've really got a great handle on this, and we can sit down with the models for climate change, and we can predict what will happen and all that kind of stuff. And that all got blown away one day. And that day was the 7th of March, 2007. We had really heavy rain upstream from our study area, just an absolutely torrential downpour that went on for several hours. Unusual, but those sorts of things are becoming more common with climate change. And so a huge wall of water came roaring down the Adelaide River. It arrived near the tributary from Fog Dam at about the same time as high tide. So the water backed up, and it backed up into our study area, and our entire floodplain went underwater for the first time, way underwater, and it drowned all the rats. So that the snake food suddenly was gone. The snakes didn't drown. I mean, they're fine, but the rats did. So these are some graphs of what happened to the pythons after that flood. So this is the number of rats we catch every year, and Thomas goes out for 2 weeks every September, and the number of rats vary from year to year, but there's always quite a few rats. But after the big flood, there were essentially no rats. Now this was a local effect. It wasn't happening right across the area because we went to a place called Beatrice Hill, which is just a couple of kilometres away, but it's high ground, so it didn't flood. And there were lots of rats on Beatrice Hill. So this was entirely driven by that flooding event due to that one day's rain. And if we look at what that did to the pythons, this is the body condition of pythons. So a high score here is a fat python, a low score is a thin python. And you can see that over the years, we had years when the pythons were fat, years when the pythons were thin, but after the rats drowned, the pythons became thinner and thinner. They became far thinner than we'd ever seen them before. They were basically, you could see the bones almost through the skin. They were just emaciated and dying. Now, again, if we go to the local hill, a couple of kilometres away, where the rats were still there, the pythons were fat and sassy. So this was entirely due to the drowning of rats, removing the food. And sadly, those snakes basically almost all starved to death. And you would go out there and find dying pythons around you. It was just awful. So one day's rain, 50 kilometres away from our study area, virtually wiped out the water pythons for the next several years. Now, this was a species that was amazingly common. We could get sometimes 20 or 30 water pythons just going up and down the dam wall once at night. And suddenly we rarely see them now. If you put the work in at night, you can find one or two, but they are a rare snake, and they're small and they're thin. So snakes are very good at surviving long periods without much food, but eventually they can't handle it anymore. And there was a double whammy. So the picture is a poor emaciated python who's actually trying to eat a road-killed snake, which they would normally never do. They're normally rat eaters, but once the rats were gone, they started eating everything they could find. Because the other thing that's happened in tropical Australia for reasons we don't understand is that the small mammals are gone. They've just disappeared. Places where you could put out live traps and catch dozens and dozens of rats and dasyurids and all kinds of things, you get nothing. And it's probably feral cats, possibly combined with wildflower, which in turn has been driven by invasive pasture grasses and maybe invasive herbivores, buffalo and cattle and so on. There's still a lot of debate about what's going on, but it means basically there is no food for a mammal-eating reptile. So the pythons of tropical Australia are in big trouble, and the water pythons have never been able to recover from that drowning event. So the conclusion, I think, is that climate change can be catastrophic, but it's not going to be, this 1.5 degree increase over X years that is the problem. It'll be the increased frequency of extreme events, like that rainfall event that we had. That makes it very hard to predict, but it's catastrophic. And other things that are happening in the ecosystem, such as this broad decline of mammals, can exacerbate that enormously. So it's a terrible story, but most stories about conservation these days are pretty awful. The second story I want to tell is about the impact of an invasive toad. This is the cane toad, giant frog from South America, brought to Australia to control beetle pests in sugarcane plantations, which has got huge toxic shoulder glands, and the poisons are distinctive to toads, and we don't have any native toads in Australia. So the Australian predators didn't evolve with those kinds of poisons around, and if they try to eat a toad, they die of a heart attack. So this toad is a giant. It's about 10 times the size of your average adult toad, but it shows you that this is one big frog. And the main victims are things like the goannas, this yellow-spotted monitor, floodplain monitor, red-bellied black snakes, and so on. If one of these guys tries to eat a toad, it'll be dead within 5 minutes. And so the toads came from South America to Puerto Rico. They were taken to Hawaii in 1932. 1935, 101 toads were brought to Australia. They were bred up. The babies were released along the eastern coast, and they have spread right across tropical Australia. Within another year or two, they will actually hit the west coast. They will have gone right across the Australian tropics. They're incredibly abundant in most areas they're found. So what does this effect have on the snakes? Well, we looked at this in some detail. So for example, one of my students, Ben Phillips, who's now an eminent professor in his own right, looked at death adders as the cane toads arrived at our study site up by Fog Dam. And we were radio tracking a lot of death adders. And about 50% of them were killed almost immediately by toads. So we'd go out looking for the signal, and we'd find a dead death adder next to a dead toad. It had bitten the toad and started to eat it and died of a heart attack. It turns out that death adders are especially vulnerable. They're an ambush predator like like American rattlesnakes and so forth. And they rely a lot on wiggling their tail tips to attract prey. And it turns out that cane toads are absolute idiots when it comes to a waggling tail tip. They're more attracted to it than the local lizards and frogs, who've had millions of years to work out that a wiggling tail tip might actually be a problem rather than a food opportunity. But the cane toads don't know that. And so they come hopping across as soon as they see that wiggling tail tip. And so they're very easy for death adders to catch. And the other problem is that death adders have got a cute trick to deal with the local frogs, many of whom have poisonous secretions in their skin. But those poisons break down as soon as the frog dies. So what the death adder does is it bites the frog, waits for about 15 minutes, and then eats the frog. And by that time, the poisons have broken down. But the poisons of cane toads don't break down. They last for months. And so the death adder waits patiently, but then it finally eats the toad, and then the snake dies very shortly thereafter. So that all sounds like bad news, but it's not all bad news, because remember, the cane toads are also killing a lot of other animals, including those big varanid lizards, those big goannas. The floodplain monitors are 2 meters long. These are some of the biggest lizards in the world, incredibly abundant before the toads arrived, and they were eating a lot of death adders. So that the death adders have been killed by eating toads, but on the other hand, there's a whole bunch of death adders that aren't being eaten by goannas now. And so most snakes have actually become more abundant since the cane toads arrived. And our best data are actually not from the death adders, but they're from carpet pythons, which I'm sure most of you will be familiar with. Carpet pythons will try to eat cane toads occasionally. They don't do it very often. If they do, you find them lying dead. You find with a big bulge in them, you cut them open, and sure enough, there's the toad there. It's obvious that a carpet python that eats a cane toad is going to die. However, if you look at the numbers of carpet pythons in our study area, my postdoc, Greg Brown, has been with me for 26 years now, and Greg walks up and down the wall of Fog Dam almost every night over that entire period, and he scores the number of snakes and frogs and so forth. And carpet pythons in this period before the toads arrived, the open histograms, were actually less common than they have been since toads arrived. So presumably escaping predation by the big varanid lizards has helped carpet pythons more than being poisoned by cane toads has hurt them. So it's a little bit counterintuitive. You find a dead snake, you think, oh, cane toads are a catastrophe for these guys. But in fact, they're a help. They're the snake's best friend. But tragically, some snake species are badly affected. So I've already mentioned king brown snakes, one of the biggest elapids. These are sort of like your indigo snakes, but they're brown and they're deadly. And they used to be relatively common. Real highlight of field work in the Northern Territory, seeing these guys. I haven't seen one for a decade up around Fog Dam. Once the toads arrived, they virtually disappeared within less than a year. So what can we do? We actually have worked out ways that we can help other vulnerable species survive the arrival of toads. And that's surprisingly, by training them. A predator that eats a small toad survives and it learns not to eat any more toads. The problem is that most of the toads at the front are large. And so if you eat one of those, you die of a heart attack. If the first toad you meet's a small one, you eat it, you get sick, you learn not to eat another toad. And so I had the idea that we could actually expose predators to small toads. So we did this with mammals to start with, these little dasyurids. And sure enough, if we train some of them on small toads so they learn not to eat them, and then we let them go, they survive. Whereas the ones we haven't trained die. It works beautifully also for northern blue-tongued skinks. The guys we train survive when the toads arrive, whereas the guys we don't train die very quickly. And most importantly, it works for these big goannas that are such a big, important apex predator on the system. Georgia Ward-Fear did a PhD on this, showing that if you train free-ranging goannas, they will survive after the toads arrive. And in a recent study, we've actually done this now at a landscape scale. So as the toad front moves through remote northwestern Australia, and this is remote countries, we work with local Indigenous communities, Aboriginal Australians, to raise toads, small toads. We released almost 250,000 small toads in advance of the toad front, and in order to try to train the predators, and it worked like a charm. So these camera trap data on the number of goannas, the solid lines show the areas where we released teacher toads to train the goannas, and in all three of those sites, goannas are still common, quite abundant. And in all the sites where we didn't release them, goannas have disappeared. So it worked remarkably well. We can train predators not to eat cane toads. The beauty of the system is that a single generation of education may be enough, because once the toads arrive and start breeding, there's lots of baby toads around, and the next generation of predators can learn for themselves. You've just got to buffer that first impact when the only toads around are the big guys that will kill you if you eat them. But sadly, that method doesn't seem to have worked very well with blue tongues or snakes. One of the problems may be that it really doesn't take a lot of toad poison to kill a snake or a lizard. So how can we teach free-ranging snakes to do it? We know that some snakes at least are capable of taste aversion, of learning not to eat something that makes you sick, but even a small toad can be fatal. So in order to train toads, we would train snakes, we would need to have a toad that was even less toxic than a small toad. So how could we do that? How could we produce a toad that had less poison? And modern molecular genetics gives us a way to do it. I don't have time to talk about CRISPR-Cas9. I reckon most of you guys will have heard of it, but you can inject a recently laid toad egg and you can knock out a particular genetic pathway. And in this case, you can knock out genes that make the toxin. And so you can create a toad that can't make a normal poison. It's much less poisonous than a normal cane toad. It's not a genetically modified organism. It's not transgenic. It doesn't contain genetic material from any other animal. It's a simple knockout, just like a mutation. So it doesn't have all of those ethical and environmental risks and so on associated with the use of genetically modified technology. It's just a simple knockout. And so we're going ahead. We haven't actually got to field deployment, but we've got the basic system working. Our first target was pigmentation. We wanted to see if it worked, so we injected guides that would knock out the genes that affect a toad's color, and sure enough, we got funny-looking tadpoles. We grew them up into albino toads. When we bred them, we had albino eggs, so it works like a charm. And we're working now on knocking out toxin pathways. And the idea is that we'll produce this animal, and it'll probably be a leucistic toad or something quite distinctive, so that when we're going to go ahead and release our 250,000 little toads, instead of releasing normal ones, wild type ones, we can release the toxin reduced ones, so that it'll be much less risky for the predators, and hopefully be a more effective way to enable some of the reptile predators to coexist with toads. So watch this space. I hope to get this up and running in the field within the next couple of years. So the third and final example is about sea snakes. And I've already mentioned at the beginning, we've got 30 odd species of sea snakes, and sea snakes are really abundant, and they're important components of tropical ocean ecosystems in the Pacific, but they have declined mysteriously across the world in many, many sites. So why? And the easy answer would be that we're trashing the ocean. So the high temperatures have led to coral bleaching, kills the coral, overfishing, water pollution, sediment accumulation. And so, you often get a beautiful site turns into a pretty horrible algal dominated site. And that's actually happened in some of our long-term study sites. I've been going back to New Caledonia every January for 20 years now, doing a population study of a small species of sea snake that occurs there. And some of our sites have degraded to the point that it's pretty obvious why there are no snakes anymore. But the really scary bit is that this same decline is happening in places that look pristine. So here's a beautiful little island called Île aux Canards, the island of ducks. It's about 1 kilometre offshore from our main study sites, which are just down here, but it doesn't get exposed to all the same problems of pollution and so on. And it's been very rigorously protected. And so if you go out there, it's beautiful coral, there's beautiful big fish swimming around everywhere. It looks absolutely terrific. But we've got data from volunteers that have been surveying this area almost every day for decades. And snakes used to be common. They used to see an average of 6 or 7 a day. By 2011, several years after they started, it was down to 1 or 2 a day. And I've actually got data for the last 10 years or so, and there's essentially close to 0. So the snakes have gone, despite the fact that the system looks terrific. So why have the snakes disappeared from Île aux Canards? And one possibility is that it's predation, that maybe the recovery of those fish has included enough big predatory fish that they eat the sea snakes. So how would you test that? It's difficult to do because predation is a hard thing to study. By definition, a prey organism only encounters, a successful predation event once in its lifetime at the very end. And there are ethical reasons why you can't just sort of shove snakes out there to get eaten by fish. So I had the idea that we could use fishing lures. They make these fishing lures that are designed to look like snakes. I think they make them mostly for bass fishing in North America. I've got no idea if they work, but we painted them to look like the local common sea snake, Amitocephalus, which are jet black, and we drag them through the water. And you can see that they look a little bit like snakes swimming, or at least I hope they look a little. I didn't think that the fish would actually fall for it, and but they did. So what we found was that the fish just went crazy, and almost every time we dragged a lure for more than a few meters through the water, it got hammered by fish of several species. Well, so that's kind of scary. It means that a small snake in that part of the world is really in big trouble. It's going to get eaten almost as soon as it appears. And so it suggests that sea snakes are in trouble for a couple of reasons. One is that habitat destruction that removes the fish and the food. But the other one is that if you protect a system well enough that all the big fish recover, then the sea snakes have got another problem. And so this is let's just keep going. So sea snakes are in trouble. They can't escape hot weather. You know, a land snake can go and sit in a cool spot underground. The sea snake can't. The water's all the same temperature. The habitat's been destroyed. Every time a sea snake has to go up to the surface to take a breath, which they have to do pretty often, they're in open water where predators can potentially get them. Whereas a land snake, of course, can just hide underground and not be exposed to predators for weeks on end. And it looks like sea snakes are only persisting in that Goldilocks zone, where disturbance has to be low enough not to have destroyed the habitat, but high enough to have knocked out some of the big fish. So they're in trouble. And we need to have that perspective as we try to work out the best ways to conserve them. So although most snake declines are probably due to a lack of prey, I think if you get rid of the frogs, the golden tree snakes are in trouble, but there will be some cases where predators are the big issue as well. So what are the implications for conservation? Some of the problems are specific to the species we look at. Cane toads, it turns out, well, some species are probably going to do fine. We don't have to worry about them. Others, we're going to have to get in there and come up with novel targeted means of management. The really scary one, though, is that many of these problems, like with the water pythons and the sea snakes, are going to require much broader global solutions. We not only need to conserve prey populations, but we need to address global climate change. And of course, that's incredibly difficult to do at a political level for all sorts of reasons. But we have to confront the fact that in many cases, these local declines are driven by global processes. So what's the prognosis? I think most species of Australian snakes are probably declining. Some of them will go extinct. There are new challenges appearing. Fire ants have recently got to Australia and they're starting to spread. But the snakes are resilient. Some of them are increasing in numbers. Red-bellied black snakes, my favourite snake, are still common in the suburbs of Sydney. If you go into agricultural lands, brown snakes, Shooternagia, are incredibly common across huge areas of Australia because people have provided the mice that they need for food. And there are some unlikely success stories, such as the big pythons. In most of the cities in Australia, there are pythons up there in the roof spaces of the houses, including my place. Diamond pythons love it there. So they have managed to thrive because people water their gardens, they encourage possums and rodents and so on, and the pythons have said thank you very much, and they move in. So what's the way forward? What can we do about it? How can we conserve Australian snakes? I think we need more research. We do need to find out what's happening. We've studied a very small number of species, and we need to be creative and think outside the box and coming up with novel approaches, including things like, the genetic modification sort of things. But I think one of the most crucial issues is that we have to involve the public. And Snake Week's a good example of exactly what we should try and do. One of the things that I've done, as Emily alluded to, is that over recent years, I've started writing lots of books designed for the public about snakes and about research on snakes and about why they're worth having around and how we should conserve them and things like that. And I see my generation of snake biologists, as they're getting older, many of us are turning to the idea that we need to talk to the public. And so that's happening more and more. Broadly, unless we can convince Australians that snakes are worth conserving, the politicians are not going to make the decisions that are needed in order to make that possible. So I think we have to get out there and we have to talk to each other and we have to talk to our neighbours and we have to explain to them why it is that these animals that are so unpopular are indeed worth having around. So look, that's the end of the story I want to tell. Work I've talked about has been done collaboratively with lots of other people. I've thrown the names of some of them down here. I thank the organisers, including Emily, the photographers, and again, I've thrown their names here. I'm a terrible photographer myself, so I've stolen photographs from other people that are far more talented. Thank you all for listening, and I'm very happy to take questions if people would like to ask them.
Thank you so much, Dr. Shine. That was wonderful and inspiring. And I personally am just blown away by the idea of the teacher toads. I think it's just so cool because I remember always thinking about how like that works for the first generation, how would it work for the second generation? But you really just articulated that really well, that process. And I think that's just really, really wonderful and visionary. So thank you. Folks, if you have questions for Dr. Shine, put them in the chat. I'm going to go ahead and moderate those now. I'm going to get started. So feel free to enter those. We have lots of time for questions. We have about 15 minutes if Dr. Shine can stay. So let's go ahead and begin. Okay. First question is, do snakes have temperature or environmental sex determination like bearded dragons or sea turtles?
So far, the snakes that we have got good data on have genetic sex determination rather than environmental sex determination. It's really mixed in some of the other groups, particularly the turtles and some of the lizards. It's very common for environmental temperature to determine the sex of an offspring. We don't know any cases where that is happening in snakes, but we have weird data from one of our fog dam species that looks as if the amount of water in the nest affects the sex of the offspring. So, we tend to think of genetic sex determination and environmental sex determination as two totally different things, but we know from several lizard species now that you can have both. You can have a system where your sex is normally determined by your chromosomes, but if you happen to develop in a place that's a bit too hot or a bit too cold, you ignore your chromosomes and you go ahead and develop as the other sex. And I suspect that we will discover eventually that lots of snakes have got exactly that same kind of complexity.
Awesome. Okay, let's see. Is there a trend in which Australian snakes are decreasing or increasing due to the cane toad invasion?
Yeah, look, there are strong differences among species. So the colubrids in general, the harmless snakes, are doing pretty well. They came to Australia relatively recently. They have close relatives, related species in Asia, where there are plenty of other toad species that have very similar poisons to cane toads. So the keelback, for example, our only natracine, that's sort of related to your garter snakes and grass snakes, they can handle toad toxins. They eat cane toads and they're fine. If they eat a really big one, they lie around for a day feeling sick, but they very rarely die. Whereas the elapid snakes are in much more trouble. Elapid snakes often eat frogs, and they evolved in Australia for a long period, and they can't handle the poison. The pythons in general are doing pretty well because most of them aren't terribly interested in frogs. And the ones that are interested in frogs seem to recognize that cane toads are different, and they don't try to eat them. So, it's the elapids, and it's especially the big elapids. It's the king browns, the red-bellied black snakes that have disappeared when the toads have arrived, and most other snakes have been okay.
And then a related question on that was about like keelback snakes that are tolerant to the poison of a cane toad. How does that happen? Do we know?
Yeah, look, it turns out that you only need really minor genetic changes to make you several tens of thousands of times more tolerant to the toxin. And people have surveyed lots of animals in South America, for example, that eat toads and discovered that they've all got those changes. Some of them are mammals, some of them are reptiles, birds. So it's evolved independently many, many times. If you are trying to make a living out of eating toads, it seems like evolution fairly rapidly creates those changes that enable you to deal with the toxin. Now, most Australian predators don't have that. But for example, if you look at varanid lizards, the big goannas, the ones in Africa and Asia are able to eat toads. But the ones in Australia that descended from those African and Asian guys have lost that ability. And so they die if they eat a toad. So it can change pretty quickly, but it only requires very small genetic changes to make you resistant to the toad toxin.
Oh, that's really cool. I did not know that. So we have a couple of questions about the teacher toad programs. So the first question was about the teacher toads. Will they potentially reproduce with the wild populations, and will that gene potentially pass on to the next generations?
Yeah, so that if we're actually looking at using gene knockouts in a variety of ways, including for toad control to try to reduce toad populations. That's the big thrust of my lab's work at the moment. Yeah, so that if we released, our 200,000 toxin-reduced baby toads, they would potentially breed. The chances are, if they bred with a wild-type toad, I suspect that knockout will be recessive and that the babies will be just as toxic as the wild-type parent. If they bred with each other, obviously, then they would have the low toxin. That would be better. So it's one of these things that makes it really hard to model what's going to happen, but the worst comes to the worst, where we'll have the same number of toads as we currently have, but a fair proportion of them will be much less poisonous. So that seems to me to be a pretty good outcome.
And then there was a related question to that about have you had any pushback from the public about the idea of increasing cane toad populations by releasing the smaller toads? And if so, have you dealt with that?
Yeah, look, it was really entertaining. So that when I first suggested it, it sounds like complete idiocy, nonsense. There was the front page of the main newspaper in Darwin, close to Fog Dam, had them saying this is the wankiest bullshit we've ever heard. So it's sort of thing they like to say in the Darwin newspaper. People were initially horrified, especially the people who were in the toad busting groups that have been trying to go out there and collect toads and kill them and get rid of them that way. And the idea of releasing more toads just seemed stupid. So what we did was we conducted a whole series of carefully controlled experiments, like the ones with the quolls, followed by the ones with the blue tongues, followed by the ones with the goannas. And we got wide publicity about those. And so we just sort of spread the word. Whenever we got an opportunity, we would talk on the local radio stations, on the TV stations. We got Indigenous Australians working with us particularly on the goanna project. That was all due to Georgia Ward-Fear, not to me. But Georgia's got really strong links with the Balangara Rangers and some of the other Indigenous groups. And so we got this real groundswell as people began to cope with it, they began to say, well, look, this is actually working. And we're releasing toads in an area that is going to be overrun with thousands of times more toads within a month or two. And nothing else we're doing is working. And so remarkably, it went from this is the most stupid idea in the world to the government authority in Western Australia has now taken over the teacher toad program, and they are breeding up and releasing these hundreds of thousands of small toads in advance of the front as it rolls through the Kimberley. So it's now become an established management method, and I have not seen a single complaint about it in the last few years. It appears to be a battle that we've won, at least in the short term.
Great. There's a question from early on that I'm guessing is from the Python study. How does the population at Beatrice Hill compare to the Fog Dam? Are they still stable, or are they also declining?
The Beatrice Hill pythons were fine for a while after that initial the drowning event and so on, but that widespread mammal decline, eventually it seemed to have spread from the east across to the west, and so the mammals disappeared. So there are no rats left on Beatrice Hill now, not because it's flooded, there's plenty of food. You look at it, you think this will be rat heaven, and there's no rats. So it's really perplexing as to why we have lost so many small mammals, but it means that Beatrice Hill is now in the same category as the rest of the landscape around it. It's not a good place to be a mammal-eating snake.
There's a fascinating question here that I also would like to know, as someone who has a lot of experience handling vipers, but not elapids. How stressful is it for you to handle such toxic snakes like king browns or taipans? How quick are they?
I mean, look, I was one of these miserable little school kids that grew up picking up red-bellied black snakes. I don't find handling elapids particularly stressful. A really big king brown would be interesting. Eastern browns are really quick, and I don't pick up eastern browns anymore. I dropped them out of my repertoire several years ago. Um, I mean, for me, they're really bowel loosening, scary animals are the vipers and the pit vipers because I'm not used to them.
It's all
So
about what you're used to. That's what I always say.
yeah, look, if you put me next to a rattlesnake, I'll be quivering. I don't know what that animal's thinking. I don't know how it's going to react. If I'm there with a king brown, I know how it's feeling. I know whether or not it's feeling sufficiently stressed out that it wants to bite me or it might bite me. And I can just move back. I can give it some space. I know what its safe area is. But you put me with a rattlesnake or a pit viper of any description, or I'm in trouble. We don't speak the same language. It's like asking for the directions to the train station if I'm in Beijing. It's just not going to work.
I love that. That's so wonderful. I suspected as much. It's all about what you're used to. It's not that any kind of snake is more stressful or scary than another. It's what we're used to. So, well, awesome. I think with that, we will conclude this presentation. Everyone, please join me in a virtual round of applause for Dr. Shine. Thank you so much for your time today, your generosity, and for all of the important work that you're doing for Australian snakes and also for snakes around the world. We just respect you and adore you so much. So thank you. It looks like Christine has her hand raised. Christine, do you want to unmic and ask a quick question?
No, I hit the wrong button.
Hi, Christine.
But,
Hi, Christine.
but I love the talk and it was amazing. I'm glad I got to meet him this way.
Thank you so much, Christine.
Okay.
Okay, well, everyone, so there's still a lot of events left for Snake Week. Definitely please follow us online, go to our website, sign up for our newsletter, and let's keep the momentum going so that we can do this every year and not just in the year of the snake. So thanks so much, everyone. Check back to our YouTube channel. We'll have these posted for you later on. Thank you, Dr. Shine.
Yeah, bye, everybody.